Dear learners, we have now come to part 3 of anti-nutritional factors. We have discussed briefly in the earlier parts of the module about anti-nutrients in foods, their classification based on specific action, and different anti-protein, anti-minerals and anti-vitamin compounds in foods. The role of each of these in interfering with the digestion and utilization of nutrients is explained. We shall now be studying on some more toxic constituents in foods and their elimination process, hitherto not included in the classification discussed in part 1 and part 2 of this module. Now, this part of the module deals with toxic constituents and their biological effects and elimination of anti-nutritional substances. Some of the anti-nutritional compounds in food with their biochemical effects are summarized in the following table 1. Let us now see the different anti-nutrients, their occurrence and biochemical effects. Now, the first one is saponins. It occurs in legumes such as soya bean, peas and beans, and the biochemical effects that are seen are it forms complexes with proteins and lipids, possesses a hemolytic effect and is known to be a gastrointestinal irritant. Now, the anti-nutrient glucosinolates, it occurs in broccoli, Brussels sprouts, cabbage, cauliflower, and soya bean contains glycosides called goitrogens. The biochemical effects that are seen are they produce toxic component isothiocyanates, then they also produce goitrogens that cause the thyroid gland to grow by inhibiting the iodine uptake. Then the third anti-nutrient we see here is amylase inhibitor. Now, amylase inhibitors are seen in cereals, that is rye, triticale and barley. Their biochemical effects show that they prevent the action of enzymes that break the glycosidic bonds of starches and other complex carbohydrates. Next, we come to tannins. Tannins are present in coffee, tea, wine, grapes, cranberries, strawberries, blueberries, apples, apricots, barley, peaches, dry fruits, mint and basil. Now, the biochemical effects seen because of the presence of tannins in food is they inhibit the activities of trypsin and chymotrypsin, amylase and lipase, then they decrease the quality of foods and they interfere with the dietary iron absorption. The next one is cyanogenic glucosides. Now, these are generally found in millet, maize, oil bean, seeds and cassava. And the known biochemical effect is they are inhibitors of cell respiration. Now, let us see how these anti-nutritional substances present in foods can be eliminated by some simple processes. Now, some of these plant chemicals have been shown to be deleterious to health or evidently advantageous to human and animal health if consumed after adequate processing. The chemical and physical characteristics of the anti-nutritional factors determine the choice of appropriate treatment that is used to eliminate the undesirable compound from foods. Number 1 is heat treatment. Heat processing is widely accepted as an effective means of inactivating the thermolabile anti-nutritional factors in food material. This improves the protein quality by inactivating anti-physiological factors, particularly trypsin inhibitor and hemagglutinins, and by unfolding the protein structure. Heat treatment processes includes boiling, autoclaving, pressure cooking, extrusion cooking and toasting. Now, boiling. Cooking generally inactivates heat sensitive antinutritive factors such as trypsin and chymotrypsin inhibitors and volatile compounds. Cooking for 60 minutes at 100 degrees centigrade was reported to be sufficient to inactivate over 90% of the trypsin inhibitor activity in food materials. The second method is autoclaving. Autoclaving under pressure for 30 minutes at 125 degrees centigrade and 15-pound pressure destroys the thermolabile inhibitory substances such as cyanogenic glycosides, saponins, terpenoids and alkaloids. Now, simple pressure cooking, that is pressure cooking under pressure for 30 minutes is known to remove trypsin inhibitors in food. Then microwave treatment. Microwave ovens use frequencies of 2.45 and a wavelength is 12.2 centimeters for 10 minutes in order to eliminate the trypsin inhibitor and the hemaagglutinating activity in food. Now, extrusion cooking is also used to eliminate the anti-nutrient factors in foods. The cooking process in this takes place within the extruder where the product produces its own friction and heat due to the pressure that is generated, that is 10 to 20 bars of pressure. The process can induce both protein denaturation and starch gelatinization as well as complete inactivation of hemagglutinins in food materials. Then coming to the next major procedure, that is without the use of heat, soaking. Soaking could be one of the processes to remove the soluble anti-nutritional factors. Seeds soaked in water at 22 degrees centigrade for a period of 18 hours were reported to decrease trypsin inhibitor activity in food. Then germination or sprouting is another procedure used to eliminate the anti-nutrient factors. Germination has been documented to be an effective treatment to remove some anti-nutritional factors in legumes by mobilizing secondary metabolic compounds which are thought to function as reserve nutrients. Germination can also lower the phytate content in legume seeds depending upon the type of bean and the germinating conditions. The structure of anti-nutritional factors and their chemical properties, especially heat lability, dictate which physical process will be more effective in their reduction or removal, thereby minimizing the adverse biological effects. Now, let us see the different treatments in use to deactivate the anti-nutritional compounds which are summarized in table 2. This table shows the bioactive substance and the commonly used elimination process. For if the bioactive substance is enzyme inhibitors, then heat treatment can be used for its elimination as we have discussed earlier. For phytic acid, enzymatic degradation, germination and fermentation can be used for eliminating its anti-nutrient activity. Now, for oxalate, cooking and dehulling are the procedures that are recommended for elimination of anti-nutrient activity. Then for phenolic compounds, dehulling, for saponins, sprouting, and for lectins, heat treatment. Now, further to this brief discussion on anti-nutrients in foods, the upcoming modules will explain in greater details the structure, mechanism of action, sources, adverse or beneficial effects and the processing techniques to make food safe for the consumer. Let us now conclude this module on anti-nutritional factors. Anti-nutritional factors are described as compounds or substances which act to reduce nutrient intake, digestion, absorption and utilization, and may produce other adverse effects. Now, the anti-nutritional factors we have seen in plants may be classified either on the basis of the structure or their actions which they bring about, or their origin. Now, the balance between beneficial and hazardous effects of plant bioactive and anti-nutrients rely on their concentration, chemical structure, time of exposure and interaction with other dietary components. Elimination of anti-nutritional factors in foods can be achieved either by selection of plant genotype with low levels of such factors, or through post-harvesting processing like germination, boiling, leaching, fermentation, extraction, etc. Knowledge regarding processing techniques to reduce the content of anti-nutritional factors in food is needed for health and well-being of the population. Technological strategies in order to obtain toxin-free products are urgently warranted.